mentioned, I am from the Pacific Northwest National Laboratory in Central Washington, Richland, Washington, and no, it's not Seattle, it's in the middle of nowhere. So I work with Dr. Janet Jansen, who's kind of a world expert in microbiomes from both gut and soil. And I've been working with Martina now for about 2 years, some form or not, looking at microbiomes related to plants. So I guess it's more of a general thing, and we'll get started. So I'll start with my personal scientific hero, and that's Galileo Galilei. And if you don't know who that is, we can talk afterwards, you should know that. So it wasn't long ago that we couldn't openly practice science. It wasn't that long ago, it was only 3 or 4 years ago, where if you would practice science, you could be burned at the stake because of the thing with the church. His personal friend Giordano Bruno was burned at the stake for believing the earth revolved around the sun. So we're all blessed now that we can be scientists in this day and age and get paid for it and not be considered heretics. So once again, he's my personal hero, and one of his great quotes that I really like, that I kind of aspire to be, is this quote here, and it's measure what is measurable and make measurable what is not so. So what does that mean? So if you have the inability to measure something, you need to develop a technology and a way, both kind of tech-wise or hardware, and in our modern age, software to measure it. If you can then measure it, then you can get at science. So that's what I try my career to do. So we're going to start off with kind of a general timeline here of DNA sequencing, which I've been lucky enough in the last decade or so to kind of witness this evolution. And on the x-axis, you have kilobase pairs per day per machine. So that's how many thousand bases we're able to sequence in the form of A, T, C, and G from DNA. And then on the y-axis, the x-axis, we have the year. And if you want more information about this, it's also a paper in Nature Protocols. And this is the rate of that over time. So the first evidence of this was published back in '77 from Fred Sanger. His first Nobel Prize was actually in protein sequencing. So he didn't think it was enough to win one, so we went off and won another in DNA sequencing, because it was easier, according to Fred Sanger. And there was another one that was going on at the same time, also was using heavy metals and stuff like this, the Maxim Gilbert type of sequencing, but the opposite chain termination basically completely defeated that. So what is that? So basically you have a DNA molecule where you have modified nucleotides that have chain terminators, so they come in with a fluorescent molecule, add to the growing strand, stop, release fluorescence, and then we can measure it. So, for example, A would come in, A would be green, and then something would be able to measure this. And this is all done on a large sequencing gel. Much later, ADI, the Applied Biosystems, were the first to basically speed this up on a capillary electrogel phoresis type of technology. And this basically fed the entire human genome project. From there, the first higher sequencing was developed, and this was done by Mustafa Ranji and Paul Rüdigen at the Royal College of Institute in Sweden. And what that was, is the idea was to have DNA sequencing that would come in, and every time the base during the polymerization occurred, you would get a small phosphate group that would be cleaved, that would then be used to power a luciferase reaction that would generate fluorescence. So that was the big difference there. Instead of chain termination, we're indirectly detecting cleavage of and incorporation of the nucleotide. From there, the first commercial version of that, 454 GS20 was done back around 2005, and that would give you around 100 base pairs and around 20 megabytes of sequence. So this was the Sanger era. This is the domination, so all the way up from the initial publication with gels, all the way up through the human genome to around 2005. Then something that was very interesting came in back around 2007. The Illumina technology came in, and I'll talk about that more in a minute. Solid, and then we had an upgrade for 454 which allows you to do higher throughput particle sequencing. Recently, around the same amount of time, around 2009, 2010, the PacBio and Oxford Nanopore were developed, and I won't get into great detail about them today, but they're up-and-coming long-read technologies. I was actually part of the process of building the molecular platform, which eventually got bought by Illumina back in 2012. And the idea there is you have a long molecule, you do dilution, and then you fragment and move forward. And then we have now the Illumina X10, which is basically 10 giant Illuminas going at once, and we're producing on average 2 terabytes of DNA-based data every week. So that's the equivalent of 30 human genomes at 30x coverage. It's so much data that it's cheaper to resequence that data than analyze it twice. And so this is now the Illumina era that we are in now currently, but I personally believe that these long-read technology, especially nanopore technology with Oxford, will eventually replace this other platform. So as I mentioned, in 2007, the Illumina sequencer comes in, and if anyone knows about Moore's Law, the Moore's Law was basically done through the Hewlett-Packard person basically said that it's based on semiconductors and the size of semiconductors and how much semiconductors you can actually pack into a computer chip, and they get infinitesimally smaller to where you have no more space to actually put any more semiconductor, and that's kind of how this law is dictated. The DNA cost per base pair actually defeated this prediction, meaning that on average the cost to raw sequence a base in a DNA sequencer is less than what's predicted by Moore's Law. This is the first time it's ever happened. This Moore's Law would predict that you would have a steady decline. So because of that, sequencing has gotten cheaper, it's gotten faster, to the point where we can't even keep up with it anymore. And so this will eventually, the one thing that will hit Moore's Law with DNA sequencing is that the storage of the data is a Moore's function. So we'll be able to sequence it cheaper and cheaper and cheaper, but we won't be able to store it. And here's some general platforms that I mentioned. It's still, it's officially extinct as of this year. The 454 Pyro sequencer is officially off the market as of this year. So we don't have to talk about it much anymore. There's the ion torrent, the ion proton, which is similar to pyro sequencing, but instead of detecting phosphorus, the pyrophosphate cleavage, and moving on from that point and generating luciferase, you take that and actually measure the pH. It's the most sensitive pH meter ever. However, this technology is similar to 454, it has indel errors, and it's expensive. MySeq is kind of a benchtop platform for Illumina, and what does Illumina do? So Illumina, basically what it does is it's much like Sanger. It comes in, you have a base that comes in, and instead of ending the chain, it actually comes off and does it against the reversible chain terminators. And so you're doing DNA sequencing by synthesis. It is the Swiss Army knife. We have it in 4 different platforms, from very, very short reads to very, very long reads, and extremely high throughput. The last 2 I mentioned here, PacBio is based on movable waveguides. So you have tethered polymerases and DNA threading through a polymerase through a pore, and you're measuring optics from the top and actually reading the DNA out. In the Oxford Nanopore, we actually have biological pores, that alpha-hemolysin pores the DNA runs through, and you actually detect individual bases as it goes across. Both currently rather expensive, but as we get higher and higher throughput and cheaper, I actually predict that it will replace the Illumina platform. So, why? Why sequence? Why? And so the reason that I got into it is because I'm interested in reading genomes. And so, and this is why. So how do we obtain genomes? So classically, up until about 2005, we only did culture them. So you have to put them on a plate, culture bacteria, and then from that point, you can then sequence their genome. And it's absolutely important that we continue to do this because they are absolutely perfect references for database searches for community samples. The next way, which I've also dabbled in, is single-cell genomics. So you basically use like flow cytometry or high throughput microfluidics You basically grab a single cell or move them into a well, you lyse the cell, you use a whole genome-based amplification, and then you directly read out that genome. The problem with this is, in principle, it's really great because you can get individuals' genomes from a population. Because even with a culture system, you're looking at a clonal population, so you're not getting the individual information from that culture. But single cells you can. The only problem is that because of all the amplification bias, the best I've ever seen is around 90% of the genome is actually covered in completeness. But the problem with that, and the only reason you get 90% with that one is because the binary fizz, and you got two microbes that are basically the same, and so you can compare the genomes. But in most cases, we're only getting 30% of the genome. But if you have a bacteria, you only see 1 in a million, 1 in a billion times, and you can get 30% of that genome. It can tell you a lot about the biology of that bacteria, because as we all know, 99% of the stuff is uncultivatable. But we can talk about what I think that means as well. And then the last one that I've kind of come into recently, because we've gotten such cheap sequence nowadays, is that we can basically do metagenomic, so shotgun sequencing of the DNA from a sample, and we can actually do this population genome binning. So the idea of actually taking metagenomic sequence and reconstructing genomes from all that data without having to culture them, without having to grab them with special equipment. So, once again, why do we need these genomes? Functional studies demand error-free genome sequence as a starting point. So, if you're trying to do multi-omics, you're trying to use proteomics, so you cannot even figure out what those spectra mean without a genome. So it's very important for proteomics, as well as transcriptomics, how they reference genomes and maps. The next one is the availability of data in organization provides unique biological insights. So what does that mean? So most people think with bacterial genomes that they're perfect circles. But I used to know a friend who worked on alpha proteid bacteria and oxygenic phototrophs. And that genome has a linear chromosome, it has a circular chromosome that has 5 plasmids. So by having that information, you can know the genome organization, you can know functional traits, and you can really tease apart the biology of that organism. The last one is that it's an absolute permanent and valuable reference. So if no one's ever been able to culture that organism before, and then you culture it, and then you sequence its genome in completeness with high quality sequence, it's an absolutely important value to the scientific community. I'll give you a perfect example. So I had this microbial map ecosystem where I had 20% of that ecosystem unknown. No idea what they are. We knew they were bacteria, but we didn't have any further classification. I was able to culture something randomly out of that community. And actually, I could fill that sequencing space by 5% of the data actually belong to that organism. So now I know that organism is important in our ecosystem. So by having that reference there, we're able to really gauge on how the system works. So why do I do this? And so the idea that I and Janet and I have is that we want to get a predictive understanding of microbiome. And over time, the only thing we can really do and look at were really simplified communities. Actually, the first genome bending study done by Gene Tyson and Joe Banfield were done in acid mice. The idea is to do lots of sequencing, and they're able to reconstruct the 2 main organisms in that ecosystem and do a lot of the biochemistry. So, but it's a simplified organism, system, and simple organism. But we've moved on to stuff in leaf cutter, human gut, and the most complicated of all, soil. On average, we have upwards of 100,000 OTUs per 0.25 grams of soil. So we have lots of diversity there. And we want to know this as far as our climate, is to understand how these microbes turn carbon over and how they function. And in the human gut, for example, IgE, an inflammatory state, we want to know how the natural microbiota functions in that disease state versus normal state. But in order to do that, we need good tools. You have to be able to measure. So again, soil is incredibly diverse. And of course, I got a checklist last night. But lots of bacteria, upwards of, and lots of bacterial species, and 95% of our food directly or indirectly comes from soil. So it's the most important ecosystem to study. My personal model system is Kansas Native Prairie. 40% of the global corn production comes from this area, these corn belts, if you will, of the United States. We had a lot of data, which was part of the Grand Challenge project, and then 26% of the continental landmass of the US is in a prairie-like ecosystem. Wheat, corn, barley, a lot of it comes from this area in the United States. And we project over the next 100 years it'll be wetter. And we have no effect on how that will affect food production in the United States. Once again, very, very diverse. So it's kind of a tenement of the microbial ecology, and is that if you want to understand an ecosystem, you must understand how those species function in that ecosystem. You cannot do anything without it. And so how do you get there? And so you have to get there through reading the DNA, and reading the DNA, putting Humpty Dumpty back together again through de novo assembly. And so there's two kind of flavors between a genome and a metagenome. So what is the genome? It's the entire collectivity of the DNA in that organism. A metagenome is the entire genetic repertoire, genetic potential of that community. So I always did a lot of puzzles when I was a kid, bored in the countryside. So a genome is a collective of that DNA, and I've represented it here as a puzzle. So you know what the puzzle looks like, you can start from the box and go from there. So sequencing reads kind of look like this most of the time when I look at that data, and I'm trying to get back to this puzzle. And so then I'm usually left with something like that, where I have lots of gaps, and we call these two pieces that connect to each other scaffolds. So there are two pieces of long DNA. So reads are pieces of in this case, of DNA, and then contigs are reads put back together. Scaffolds are pieces of those DNAs that we think belong with each other, but we don't really know, so we leave them as that. And then the next thing is repeats. So, in your puzzle, parts of the ocean would be repeats, because you don't know if they belong here or if they belong there. We don't know. So, in a metagenome, a community sample, so grabbing some DNA from dirt or from human gut or from a map, it is literally, we don't have a picture. We have no idea, a priori, the abundance of organisms or who's there. Not a clue. So once again, we get our reads, and we try to put it back together, and once again, we're left with a map again, but we have no idea whether they're 1, 2, 10, 100, whether they're phyla, whether they're genus-related, species-related, we have no clue. It's literally, that's why I like the anagram here, it's literally the black box of microbial ecology. So the other thing that's really kind of thing now is, like I said, you're able to take these contigs or these pieces from a mixture and put them together to get individual genomes from a mixture. And that's called metagenomic binning, or population binning is another term for it. And so this is basically a really good thing here. You have all these, we'll say, pieces of Legos or contigs, and you're trying to then put those back together. So this is called binning. And so what you want to do is be able to put all the red contigs and all the green contigs separately, because in this case, we say, well, that's one unique genome, that's another unique genome, and that's another unique genome. So how do you do that? So the first thing you look at is GC content, because usually if it's from the same genome, it has relatively the same GC content. It's problematic because of original gene transfer and vertical gene transfer in bacteria that have regions that have different GC content, making those contents fall out of your bins. Next thing is to look at abundance and coverage. So you take your long pieces of DNA that have been reconstructed as contigs, you map your reads to them, and you look at the abundance of those reads across. And that differential coverage allows you to gauge the abundance of that in a particular gene or that contig across a collection. Next thing is checking tetranucleotide frequency, which is basically a former. So you take your contigs, you count every 4 nucleotides, and that pattern is the same, reliably across phyla, and even reliably across species. So you can get asked what that organism is, just basically on like content. You can look at composition of those contexts. So if you have alpha-proteid bacteria and another alpha-proteid bacteria contig, and the tetranucleotide frequency is correct, GC content is correct, and reads map across those contigs, then you know it's from the same organism. And then usually we use a maximum expectation likelihood mechanism. to actually put this all together, and we can talk about the math if you're interested down the street. So, as I mentioned, you have this puzzle, so metagenomics, why doesn't it perform the same? Right? Because they're all a bunch of genomes, why doesn't it perform the same? And it's, as I continue on, discover why it's tough. So, high number of types and sizes of genome presence and sparse sampling leading to low coverage. So, what does that mean? So, you go and you take a sample from the soil system, and you may have 10 coli, you may have 10 Bacteroides, you might have 1 alpha proteobacteria, and you may have 1 TM7 for every 10,000 of everything else. But you're really interested in TM7, so you never maintain enough coverage to get that micro. So that's why you usually try to combine single cell plus metagenomics to get at those rare guys. Next one is that it's a lot of data usually to get there. On average, I don't really want to start any project until you have at least 100 gigabytes of data for a little. That's why those long reads are really helpful, because if you have 100 PA stretches or longer, those genomes are going to come together a lot easier. So also because there's short data, it requires a lot of RAM to actually put those reads into contigs and put them back together. And so usually you need a large cluster. I'm lucky enough at TU to have a 2 terabytes working memory cluster to actually put very, very large metagenomes together on the orders of 300 to 400 gigabytes of data. So further problems with not only with metagenomics, just that you have a lot of data, it's even more complicated when you're looking at soil. And so the last thing is that, and this is from Eric Womack, and this was back in 2008, and he was, the Nostradamus of predicting that this would be a problem. And he basically said that log short reads are not an appropriate tool. You just shouldn't use them. Well, we're going to try anyway. And so because you have short reads, it's hard to basically assign them functionally as well as taxonomically. So you need assembly to then put them back together. My favorite is from C. Titus Brown, and it's from his blog post, and if you ever want to read interesting blogs on what's going on in that guy's head, you can read his blog post. And it basically says that he proceeded to fail repeatedly at assembly for 2 years. So he got large amounts of data, part of the Grand Challenge project from the JPI, and could not put these things together. And so he developed some novel mechanisms and software how to get there, but just everything he tried couldn't get it to work. And then the last one, when they finally did get it to work, back in 2014, from Nina Howe's work, they took around 250 gigabytes, 3.3 billion 80 base per reads, that's the equivalent of 30 human genomes, and they assembled one piece of DNA bigger than 10 kb, and only 9.5% of those reads actually mapped back. So a lot of the reads mapped to nothing, and that's bad something when you're trying to do. And at this point, if people would have asked me if you could actually get genomes from soil, I would have called you a madman, because with this amount of data, it's impossible. And so what if we could obtain longer contigs and improve metagenomic characterization? So once again, how do you find, when I think of this as a process, how do you find one book, your genome of interest, read and write it down in a mixture of all the possible books in an entire library? And that's the challenge we ask of metagenomic data in any ecosystem, and it's even harder when you look at a soil where you have hundreds of thousands of organisms. So there's 2 ways, and this gets a little techy. So of course, how am I doing? Going right, nice speed, everybody? Everybody understanding? Or you're all deer in headlights and don't know what's going on? It's okay. It's okay. You can say, look, we can slow down. If any questions, any thoughts, comments? Should I continue to ramble on? Okay. So there's 2 computational approaches on how to get there for algorithms that the metagenomic assembler uses. And so the first one is this idea that was done by C. Titus, which is called kind of divide and conquer. And so his thought there was that you had a lot of the same thing over and over again that had errors that you could digitally normalize and partition those reads out and remove them and not lose any of the abundance information in order to do the assembly. And they were successful in doing that, and that was also done in a Bruin graph. So you can divide and conquer, and then you see the Bruin graph assembly when it's meta array to get there. And once again, very few contigs and very low mapping. So you can't bin genomes with this method. And then the next one, which is kind of absolutely revolutionary, which is this idea of storing threads in distinct clusters and then using de novo assembly from that point. Also using graph-based room graphs, but Metagenomic was changed. It changed the game. It was a game changer for metagenomic assembly. And from that same data, not even 2 years prior, we were able to take that and get 10 times more and get 10 times more mapping. Still not good enough, because no genomes can be binned with this, and only a few have been done in the Holtman paper with Janet in permafrost. And then we don't have full length 16S, so we really don't know what's really in silica, because you don't have full length, you can't do phylogeny, you can't really figure out what's going on. And so when I joined Janet's group, the idea was like, what's the best way to put them together? And so what I want you to think about is speed. What I want you to think about is speed, because if you had the best